Okay, so starting with our topic. So first of all, hold on. Before we get into cardiovascular diseases, let's actually go ahead and do a little bit of a revision of what we know about the cardiovascular system. In fact, hold on. Let me open up a diagram of the heart and let's quickly do a revision of what we have already done in our IG. So, all right, here we go. All right. So, we know first of all that in the human body, we have, or generally not in just human body, in mammals, we generally have a double circulatory system. So, a double circulatory system is when the blood is going to pass through the heart twice, right? In one cycle, in one circuit, the blood is going to pass through the heart twice. The first time it is going to go ahead and pass through the heart, it's going to be when the blood is going to the lungs. So one part of the double circulation is when the blood is being pumped to the lungs. And the second part of the circulation is when the blood is being pumped to the rest of the body. So the part of the circulation where the blood is being pumped to the lungs, we call it pulmonary circulation. Pulmonary circulation. And the part of the circulation where you're pumping the blood to rest of the body, that is basically known as your systemic circulation. So we know that the advantages of double circulation are generally seen when you look at larger organisms. For example, inside a fish is such a small organism that it doesn't need a double circulatory system. But we humans or mammals generally, large organisms need a double circulatory system. This is first of all to the reason for that is that you have a larger organism, and because you have a larger organism you have to pump the blood over a vast area, which is why the blood needs to be pumped with a lot of pressure, with a lot of force. So that is one of the reason why the blood is coming back from the lungs to the heart, and then the heart is pumping it again, so that when the heart is going to pump it again, it's going to have that high pressure, so the blood can travel to all the areas of the body. Secondly, larger organisms also have a higher metabolic rate, which means that they have more demand for glucose, oxygen, nutrients, and all that, which is why the blood you which is why you need to have a double circulate system to make sure that you can pump the blood fast, you can pump the blood to all areas of the body, and you can do it quickly, so you can meet those metabolic demands of the body. So those are the advantages of double circulatory system. Now, generally going over our human heart. So when we are talking about the human heart, I'm not going to go ahead and go over the structure of the heart. We already have a good enough idea of that. We know that the heart has four chambers. The first chamber on the top is known as the atrium. The chamber at the bottom are known as your ventricle. So we have right atrium, left atrium, right ventricle, left ventricle. Talking about our other important parts of the heart, we have our vena cava. By the way, we have two branches of vena cava. We have an inferior branch and a superior branch. So we have inferior vena cava and superior vena cava. Inferior vena cava is going to bring the blood from the rest of the body to the heart. Superior vena cava is going to bring the blood from the head and neck. So superior vena cava is bringing the blood from head and neck. The inferior vena cava is going to bring the blood from the rest of the body, and all of this is going to come and it is basically going to return back to the heart. Now, once the blood has entered the heart, now let's go over our cardiac cycle. Once the blood has entered into the heart, it will enter first into the right atrium. After it enters into the right atrium, what does the right atrium do? Right atrium will contract. the right atrium will contract and the blood is going to pass on to the right ventricle. That is step number 1. After that, the right ventricle has to contract. But before the right ventricle can contract, the valve has to close. So we have a valve between the atrium and the ventricle, this valve over here. The valve, general terminology for the valve is atrioventricular valve. So you have an atrioventricular valve that is present between your atrium and your ventricle. It's a general terminology. Then you have specific names at the right side and left side. So the atrioventricular valve is present, and your atrioventricular valve is basically going to allow the blood to travel. It's basically going to prevent backflow of blood. So when the ventricle is about to contract, the atrioventricular valve is going to close. The atrioventricular valve is going to close so that the ventricle, when it contracts, the blood does not flow back into the right atrium. The blood does not flow back into the right atrium. So we have the atrioventricular valve, and that atrioventricular valve is going to go ahead and it is going to close. After that, what is happening? Okay, so the ventricle is contracting. If the ventricle is contracting, the blood has to go somewhere. It can't go back to the right atrium, but it has to go somewhere. Where does it end up going? It ends up going into the pulmonary artery. It ends up going to the pulmonary artery, and pulmonary artery, even though it says artery, it carries deoxygenated blood, and it will carry that deoxygenated blood to the lungs. But before the blood can enter into the pulmonary artery, once again, we have a valve. That valve has to open. If the valve will open, only then the blood will be able to enter into the pulmonary artery. This valve is known as your semilunar valve. So the semilunar valve has to open so the blood can travel from the right ventricle into the pulmonary artery. And once the blood is in the pulmonary artery, the valve will close again. The semilunar valve has to close again so that the blood keeps on traveling in the forward direction, that is towards the lung. The blood will pass on to the lung, and when it is going to end up passing on to the lung, when it is going to end up passing on to the lung, what is going to happen? Of course, deoxygenated blood will combine with oxygen, and the blood is going to get oxygenated. Once the blood is oxygenated, it will go ahead and it will come back to the heart, and it will come back to the heart through the pulmonary vein. Now the blood is back in the heart. From the pulmonary vein, it will enter into the left atrium. Once it will enter into the left atrium will contract. You again have the atrioventricular valve. You again have an atrioventricular valve in the middle. So first, the atrioventricular valve will open. So when the atrium contracts, the blood can simply pass on to the ventricle. Once the ventricle is filled, once the ventricle has the blood inside of it, your atrioventricular valve will close again to prevent the backflow of blood, to make sure that the blood does not flow back into the atrium, and your semilunar valve will open. So in the pulmonary artery, this is the pulmonary artery, and in the aorta, in both of these, the valves are known as semilunar valves. So the semilunar valve of the aorta will open. So AV valve is closed, atrioventricular valve is closed, semilunar valve is open. So then when the ventricle will contract, all of the blood will pass on to the aorta. And once all of the blood has passed on to the aorta, the semilunar valves will close again, so the blood cannot flow back. And after that, the blood is going to be carried on to rest of the heart. This is basically what we know about our, this is our cardiac cycle. So any confusion so far what the cardiac cycle actually is? Do we need to go over it again?
No, ma'am.
No.
All right. Now, in the middle of the heart, we have a septum. What is the function of septum? To separate the right side and the left side of the heart. So septum is simply present to separate the right side and left side of the heart. Okay. The heart is also the muscle. So the wall of the heart is made of muscle. All right. So the muscle that is present inside the heart is known as cardiac muscle. Cardiac muscle is different than other types of muscles. For example, okay, generally speaking, there are three types of muscles in our body. We have skeletal muscles. Those would be your biceps, triceps, your quads. All of those are going to come under your skeletal muscles. Then we have our smooth muscles. So smooth muscles are present, for example, inside your blood vessels or inside your digestive tract or your sphincters inside your body. They're generally made of smooth muscle. And then we have cardiac muscle, which is of course only found inside the heart. So cardiac muscle is a bit special in a way that it can contract and relax without, tiring out or without fatiguing. Talking about skeletal muscle, we know skeletal muscles can only contract and relax up to a certain limit. After that, your muscles get tired. Cardiac muscle has the ability that it can undergo constant contraction relaxation without tiring out. And you will study more about this in your, I think, next year. Next year in your syllabus, you will actually study more in detail about how the cardiac muscle actually works. So cardiac muscle also has coronary vessels. So coronary vessels are the special blood vessels that bring blood to the muscles of the heart. So the muscles of the heart have their own specific blood supply, and that comes from the coronary blood vessels. So that is why if there is any disease in these blood vessels, we call it coronary heart disease, because the name of the blood vessels that are supplying the heart are known as coronary vessels. And okay, another thing. So far we know that we have hemoglobin. We know perfectly well that we have something that is known as hemoglobin, right? Now, when we are talking about our hemoglobin, we know hemoglobin is responsible for transport of oxygen. Now we're going to learn of something else that is known as myoglobin. Myoglobin is generally found in your muscles, and it has greater affinity for oxygen. That means it has a greater binding capacity or binding ability for oxygen. I mean, if you compare hemoglobin and myoglobin. So first of all, both of them are globin. Hemoglobin, myoglobin. We know globin is a term for proteins, globular proteins. We have done biological molecules. They both are your globular proteins. But myoglobin has greater binding affinity or greater binding power when it comes to oxygen. Hemoglobin does not bind that strongly, and that is what we need. We don't want hemoglobin to bind to oxygen too strongly, because when we want that, when the blood reaches the the tissues of the body or the cells of the body, the oxygen will quickly break away and go to the cells. So we don't want hemoglobin to bind to oxygen that much. But myoglobin has the greater binding affinity when it comes to binding with oxygen. So in your heart, you have a lot of myoglobin. And why do we have so much myoglobin in the heart? Because this myoglobin stores energy, oxygen. So it's stored because it has more binding capacity. It can bind to the oxygen more strongly. It allows the heart to store oxygen so that the heart can keep on contracting regularly. It can keep on contracting on a regular basis. Now, little more things we have to go ahead and go over. Your atrioventricular valve is the general terminology for the valve you find between atrium and ventricle. They have specific names as well. On the right side of the heart, it is known as tricuspid valve. On the left side of the heart, it is known as a bicuspid valve, or another terminology for that is mitral valve. So if you're generally, for example, you want to talk about both of the valves without specifying right side or left side, you can just say atrioventricular valve. You'll be completely correct. But if you want to be more specific, which valve you're talking about, so then you can either say tricuspid, bicuspid, or you can go ahead and say that it is a mitral valve. Tricuspid, the reason we call it tricuspid is because it has got three flaps. The valve has got three flaps. 1, 2, 3. The bicuspid, we call it bicuspid because it has only two flaps. And these valves, how are they attached to the heart? They are attached to the heart by tendons. You basically have these cords, and you have these cords that sort of look like this one. Clear this out. You have these cords that sort of look like this. Okay. We call these our tendons, or we call them tendinous cords because they are made up of tendons, and these are basically the ones that are responsible for opening or closing the valve. So they make sure that the valve is going to, for example, if you're closing the valve stays closed. Okay, if these cords were not present, what would happen? There's a chance that the valve would flip to the other side, or the valve would get overturned because of the pressure, high pressure inside the heart. The valve would end up getting overturned. So these cords make sure that the valves do not get overturned. They can withstand the pressure, and they can stay open or closed no matter what the situation is. Another point that we have to mention is, one thing we can clearly notice is that the left ventricle, look at the wall of the left ventricle and compare it to the wall of the right ventricle. The muscular wall of the left ventricle is much thicker than the muscular wall of the right ventricle. The reasoning behind that is when the left ventricle contracts, it is pushing the blood through the entire body. The right ventricle only has to push it up to the lungs. The left ventricle has to push it into the entire body. So it needs to have more muscles so it can pump the blood with more force and more pressure so that the blood can actually be carried throughout the entire body. Any confusion in any of this? Is this all clear?
Yes, ma'am. All clear.
All right. Now, when we talk about, now this was just a general detail of what happens in the cardiac cycle. Now we have to go ahead and go over special terminologies. Okay, so everything we discussed so far that has special terminology. There are two terminologies I want you guys to know: systole and diastole. We have systole and we have diastole. Systole means contraction. Diastole means relaxation. That's the general terminology and general meaning of it. Systole means contraction. Diastole means relaxation. So for example, what if I say atrial systole? If I say atrial systole, you will say the atrium is contracting. What if I say ventricular systole? So you will say that means that the ventricle is contracting. What if I say atrial diastole? So you will say the atrium is relaxing. And if I say ventricular diastole, that means that now the ventricle is relaxing. So that is basically this altogether is known as your cardiac cycle, because we know our heart is contracting, relaxing. So going over it again, the blood enters into the right atrium and the left atrium. So in our body, of course, both the sides are going to be working at the same time. So when the blood is going to enter into the right atrium, and when there will be blood coming into the left atrium, both the atria are going to contract simultaneously. That will be known as atrial systole. All right. After atrial systole is complete, the blood is going to pass onto the ventricle. So now the atria are going to relax. Now they're going to be like, okay, we have contracted, we have pushed the blood into the ventricle. They will relax. That will be known as atrial diastole. Okay. Now, after that, once again, our valves will close, both of them. Tricuspid, bicuspid, because both the right side and the left side work together, right? So bicuspid will close and the tricuspid will also close. And your semilunar valves will open in the pulmonary artery and in the aorta. The ventricle is going to contract both the side, right ventricle and left ventricle. This will be known as ventricular systole. Both of them are going to contract. The blood will go in from the right side, it will go to the pulmonary artery. From the left side, it will go to the aorta. Your semilunar valves are going to close. And when your semilunar valves are going to close, once all the blood is gone from the ventricle is going to relax, which is going to be known as ventricular diastole. So this is how the cardiac cycle happens. Atrial systole, so the atria are contracting, then atrial diastole, then ventricular systole, then ventricular diastole. And this all together, 4 phases are known as your entire cardiac cycle. So these are certain terminologies that I don't think you guys did in your IG. So for the contraction and relaxation, we have certain terminologies. So this is all clear, right?
Yes, ma'am.
All right. So when they ask you what is a cardiac cycle, you will say cardiac cycle is the cycle of systole and diastole in the heart. So cardiac of cycle of systole and diastole, that means cycle of contraction, relaxation, that is going to be known as our cardiac cycle. So that is a general idea of how the human heart is actually going to be functioning. Now, what else do we need to do? And what else do we have to go ahead and do in our syllabus? So this is the first thing actually you guys need in your syllabus. One of the things is, of course, first of all, the entire cardiovascular system, and then you have to go over the diseases. So the first thing you had to do was the heart. Now, after the heart, you guys in your cardiovascular system, we have two separate, basically cardiovascular system, if you talk about it, it has 3 separate components, okay? Your cardiovascular system includes your heart, it includes your blood vessels, and it includes the blood. So we need to go over the blood and we need to go over the blood vessels as well. So let's go ahead and talk about that. Hopefully the part related to heart is all clear. Just give me 1 second. I'll share the screen so we can go over our blood. All right, now going over the topic of the blood. So we have to talk about the different components of blood, which we already do have an idea of. So we will just do the same thing as IG, but in a bit more detail now. We'll study some extra things that you need to know. And we're going to talk about the role of different components. So when you are going to talk about it, what is blood? Blood is basically the transport medium, and it is a transport medium. That is basically what blood is. It is transporting oxygen, glucose, carbon dioxide, urea, your nutrients, amino acids, hormones. All of these things are passing through the blood. So it's acting as a transport medium, and its passage through the vessels or its movement through the vessel is what we call circulation. the blood will travel, or it will take the materials to all parts of the body. So for example, it is going to carry your hormones, it's going to carry your oxygen, it's going to carry glucose and all those things to different parts of the body, and then it will remove the waste products. Carbon dioxide and urea are the main waste products. It will remove those waste products and bring them back so they can be removed from the body. So let's go ahead and now talk about that. So in our component of the blood has, first of all, let's talk about the cells. Okay, the cells inside the blood, there are three major cells: red blood cells, white blood cells, and platelets. All right, then that is the cell portion of the blood. Then we have a liquid portion of the blood. The liquid portion of the blood is going to be known as your plasma. Now, when we are going to talk about it, plasma, like I already told you, it is the fluid part of your blood, the liquid part of your blood. And it is basically talking about it, plasma is not red in color. Plasma is actually yellow in color. It's the color of straw. Like the straw that you feed animals, hay, it is that sort of a color. So it has a yellowish color. The only reason it looks red is because of your hemoglobin. So plasma actually has a straw color, sort of straw color, a yellow color, and your plasma is the liquid portion, and its main function is that it is going to be carrying all the nutrients and the waste products that have to be transferred throughout the entire body. What are the components of plasma, or what can you actually find in the plasma? So actually a major portion of the plasma is just water. To be honest, a major portion of your plasma is going to be just your water. Other than that, it carries your nutrients. Okay, so your nutrients, for example, would include glucose, amino acids, it would include your lipids, it would include certain vitamins, it would include minerals, it would include ion, sodium ion, potassium ion. All of these are going to be carried inside the plasma. So the plasma is going to carry these nutrients and everything to different parts of the body. Once the blood reaches different parts of the body, all the nutrients will be provided to the cell, and the waste products of the cell are going to then enter into the plasma. So the plasma can then carry them away so they can be excreted. Two major waste products, like I told you, carbon dioxide and urea. So carbon dioxide is carried in the plasma. Oxygen is carried in the red blood cells. Carbon dioxide is carried in the plasma, and urea is also going to be carried in the plasma. The carbon dioxide will be taken to the lungs so that it can be breathed out, while your urea is going to be carried to the kidneys, and then it is excreted out in urine. Other than that, it is also responsible for transporting a lot of your hormones. You have, for example, your insulin, glucagon, right? Major hormones of the body. These hormones are going to be transported in the blood. Then it is also responsible for maintaining body temperature. Because your blood allows you to maintain body temperature, because let's say you are really in a really hot atmosphere or really hot environment, your body needs to stay cool. So your blood is going to allow the heat to diffuse out. It will allow the heat to diffuse out of it. As a result, the internal temperature can be kept cool. The opposite will happen in in cold weather. Another thing is that it has a special buffering system. So we have a bicarbonate buffering system, we have a phosphate buffering system. So there are different sort of buffering systems present inside the plasma, and this ensures that there are going to be no pH changes. So pH changes do take place inside our body generally. For example, too much carbon dioxide in the body, too much carbon dioxide would end up changing the pH a bit towards the acidic side. So pH changes do end up happening, but we have a buffering system that allows us to regulate it and make sure that the pH of the blood does not change. That was what we had to know about plasma. That was pretty much everything what we had done in IG as well. Now we go into a little bit of something that you guys might not have done before. Red blood cells. The medical name for red blood cells is erythrocytes. Erythrocytes is the medical terminology for red blood cells. And generally speaking, so the shape of red blood cells is that they are going to be a biconcave disc shape. Let's first of all go ahead and talk about why they have a biconcave disc shape. One reason is to increase the surface area. It increases the surface area so oxygen can, quickly diffuse in and out of the cell. By the way, when we're talking about diffusion and later on when we do the topic of respiration, I will explain this to you guys in more detail. But generally, when we talk about surface area, so inside our body, the terminology we use is surface area to volume ratio. What is surface area to volume ratio, right? When we are going to talk about it, let's take the example of two cubes. We have this cube and we have this cube. This is 1 centimeter, 1 centimeter. This is 3 centimeter and 3 centimeter. So what happens if I ask you guys, can you calculate the surface area of the cubes? To calculate the surface area of the cube, you're going to, first of all, find out the area. Area of the cube is going to be just 1. 1 multiplied by 1 is 1. Side into side will give you the area. And then you multiply it by the number of sides. So a cube has 6 sides. So you multiply it by 6, you get the answer of 6. So 6 centimeters square, for example, is the surface area of the smaller cube. The bigger cube is 3 by 3. So 3 multiplied by 3 gives you 9. That is the area of one side. And since there are 6 sides of the cube, multiplying that by 6 is going to go ahead and it is going to give you your, it is going to go ahead and give you the answer of, I think, 54 centimeters square. So that is how you're going to go ahead and you're going to get your surface area. Once you have your surface area, then let's calculate the volume. For volume, we always do side into side into side. So side of the cube is 1, 1, 1, 1. All the sides are equal in a cube, right? So the volume of the smaller cube is going to be 1 multiplied by 1 multiplied by 1, which gives us the volume of 1 centimeter cube. The bigger cube, on the other hand, to find out the volume, we will do side into side into side. So 3 into 3 into 3, which gives us the answer of 27 centimeter cube. And now let's take out the ratio. Let's take out the ratio of the surface area and the volume. So surface area of the smaller cube is 6. Volume is 1, which gives us ratio of 6. The surface area of the cube is 54. And this is 27. The volume. So we get the answer of 2. So the bigger cube actually, if you take out the ratio of surface area and volume, the bigger cube actually gives you a lower ratio. The smaller cube gives you a larger ratio. So in the bigger cube, because the ratio is less, the diffusion is going to be faster. In the smaller cube, since the ratio is higher, the diffusion is going to be lesser. So far up till IG, we just talked about surface area. We said, oh, it has a bigger surface area, so it's going to have faster diffusion. Now at our A levels, we're going to be a bit more specific. We don't just look at what the surface area is, we always compare it to the volume. So since the ratio of surface area and volume is going to be less, that's going to allow fast diffusion because the volume is less, but the surface area is more. Is this making sense?
Yeah. Yes, ma'am.
So when you guys are writing down about your biconcave disc, you're talking about your red blood cells, you will say that the red blood cells have a large surface area to, they have a large surface area, or they basically, when you're talking about the ratio, you are going to go ahead and you're going to say that since they have okay, I'm sorry, I said one thing the other way around. I'm very sorry. When the ratio is large, that will mean fast diffusion. I'm sorry, I said when the ratio is less. Let me just make that clear. I said it the other way around. When the ratio is large, that will mean fast diffusion. When the ratio is small, that's going to mean that's going to mean um, slower diffusion. That is why when we cut up pieces, or for example, in chemistry, we say that when we turn it into a powder, that increases the surface area. What that actually does is that it increases the surface area to volume ratio. So that actually increase in ratio is why your smaller particles give you faster reactions as compared to one large particle. So I said it the other way around. Larger ratio is going to mean faster diffusion, smaller ratio is going to mean less diffusion. So smaller block is going to give you faster diffusion, bigger block is going to give you less diffusion. So in our red blood cells, the ratio of surface area to volume is high, which is why the oxygen diffusion can take place very rapidly. That is one advantage of having this particular biconcave disc shape. The other advantage is that it allows the red blood cells to squeeze through the capillaries. They can squeeze through the capillaries. That is why when you have other blood diseases, for example, like sickle cell anemia. In sickle cell anemia, the shape of the red blood cell changes into a sickle because of the hemoglobin. And we did a concept of this when we did biological molecules. We went over sickle cell anemia a bit. So the hemoglobin changes, and because of the change in hemoglobin, your cell ends up getting a sickle shape. So the problem with the sickle shape is that they can't pass through the capillaries, so they end up getting damaged, or they end up damaging the capillaries, and that causes anemia. So the biconcave disc shape allows them to squeeze through the arteries or capillaries. So that is one more advantage that you have of the biconcave disc shape. The second thing that we need to know about red blood cells is red blood cells do not have a nucleus. Mature red blood cells. Let me be very clear, mature. When you talk about immature red blood cells, baby cells, immature red blood cells that will end up growing into a proper red blood cell. So immature red blood cells do have a nucleus, but a proper mature red blood cells do not have a nucleus. Why? So that they have more space available for hemoglobin, so they can carry more hemoglobin and they can transport the hemoglobin, transport the oxygen in greater quantities throughout the entire body. So they don't have a nucleus, mature red blood cells. But that also comes with a drawback. The drawback it comes with is without a nucleus to keep control over them, to keep watching over them, they cannot survive for long. They live approximately 120 days or 4 months. That's it. They live for 120 days or 4 months because they do not have a nucleus. And after that, they're going to be destroyed by the body. The hemoglobin is going to, the iron in the red blood cell is going to be reused. Iron is generally not excreted. It is excreted in very, very small quantities. Iron inside the hemoglobin is generally recycled. The hemoglobin is broken down into different compounds, into different pigments, and then those pigments are excreted through the bile. So when you talk about your gallbladder, we know it has bile inside of it. So one of the components of bile is your breakdown products of hemoglobin. So when you break down the hemoglobin, the products you get, they are excreted in through the bile, and then they pass out in the fecal matter, and that is how the hemoglobin is removed. Iron is taken back, generally recycled, hemoglobin is removed, and after 120 days, new red blood cells are going to be made throughout the entire body. Red blood cells are produced by the bone marrow. So we have to remember that. Okay, now let's go ahead and talk about our leukocytes. Leukocytes is white blood cells. So erythrocytes is the medical terminology for red blood cells. Leukocytes is the medical terminology for your white blood cells. And generally speaking, your leukocytes are much larger than erythrocytes. Size-wise, they are much larger. Talking about quantity, okay? So approximately red blood cells are around 4 to 5 million in females and 5 to 6 million in males. Leukocytes are much lesser. They are 4,000 to 11,000. So there are 4,000 to 11,000. Those are in millions, 4 to 5 and 5 to 6. And now when we talk about where they are made in the bone marrow. So, okay, let's first of all go over a little bit about the type of leukocytes, okay? So we have when you talk about leukocytes, we have two types. We have what we call our phagocytes, and we have what we call lymphocytes. So in phagocytes, we have different types, okay? We have macrophages, we have neutrophils, we have eosinophils. There are lots of different types of them. I'm not getting into that because that is the topic of immunity, and that we don't have to, get into all of that right now, but we are going to do, not right now, in next year when you guys are going to be doing your A levels. In that, you're going to go ahead and study quite a lot of details how the immune system works. So we have our phagocytes, which are responsible for digesting, for engulfing the bacteria, and then for destroying them. Then we have our lymphocytes. Lymphocytes are of two types: B cells and T cells. B cells are the ones that produce antibodies. So we have been hearing, especially in COVID, antibodies, antibodies, antibodies. So B cells are the one that actually make antibodies. And then the T cells are present that actually do their job in other mechanism. They do their job with some other mechanism. So generally, all of these are initially going to be made in the bone marrow, all of them. All leukocytes are initially made in the bone marrow, but the T cells, what happens is that they will be made in the bone marrow, but they don't stay in the bone marrow. They end up going towards the thymus. Thymus is a small organ that you find in your throat. So it's present in your throat, and it is a very small organ, pretty small. And so the T cells actually, they will go to the thymus, and then they will mature in the thymus. So your lymphocytes, B cells, your phagocytes, they will all stay in the bone marrow and they will mature in the bone marrow. But T cells are going to go to your thymus instead, and that is where they're going to be maturing instead. So when you are talking about the structure of it, generally the structure of, first of all, your phagocytes, okay? So your phagocytes, generally, they have a large shape and the nucleus is lobed. Lobed meaning that, it looks like this. It looks like it's got multiple projections or multiple lobes, right? So the nucleus of phagocyte is generally lobed, and the nucleus of your lymphocyte is big and circular. So lymphocytes have a big and circular nucleus. The other ones have more of a lobe nucleus. And of course, we know their function. Their function is for immunity, for defense. They're going to be coming and doing their multiple jobs. For example, phagocytes are going to be engulfing and eating the bacteria. B cells are going to be producing antibodies. T cells are not only going to be controlling the entire system, or they're not only going to be helping and stimulating more and more cells to come and do their job, they also produce certain chemicals that help the process happen much more better. So that is what you have to know about your leukocytes. Then we have our platelets. Platelets are what? Platelets are generally, so in our body, what platelets are generally first produced as megakaryocytes. That is like the initial form of platelets. So platelets are first made as large cells that we call megakaryocytes. And then these large cells are sort of broken down into small fragments. And those small fragments are going to be known as your platelets. In quantity, they're about 150,000 to 400,000. So approximately from your 150,000 to 400,000, that is the amount of platelets that you're going to find in the blood. And platelets are generally involved in the process of clotting. I'm not going into clotting right now because we do have to do clotting later on in this same chapter. We have to know the process of clotting in good detail. So when we go over that, I'm going to discuss with you step by step how the clotting is actually going to take place. Any confusion so far? Does anybody want to ask anything?
Ma'am, everything is clear, but can you repeat that when hemoglobin was broken down?
Sorry, Basit, what can I repeat? Can you say it again?
Ma'am, when hemoglobin was broken down by bile or...
Yeah. Okay. So when you're talking about your hemoglobin, so inside your red blood cells, you have your hemoglobin, which is a protein, and that hemoglobin is attached to your iron. And when we did biological molecules, we said that these sort of proteins are known as conjugated proteins. Now, the iron, first of all, it is generally not excreted. It is excreted in really small quantities, but most of the iron is generally recycled. Since iron is excreted in small quantities, so that is why we need it in our diet to make up for the small quantity that is being excreted. But most of the iron is generally going to be recycled in our body. Now, when we are going to talk about hemoglobin, hemoglobin is broken down into pigmented compounds. Pigmented mean that the compounds of hemoglobin, when you break down hemoglobins, you break down into different components, right? I'm not getting into that because that is another chapter that I think we have to do it not this year. Next year, I think we'll do this in more detail. But basically, it is broken down into a lot of different products. After the hemoglobin has been broken down into different products, it has to be removed from the body. So the liver is the one who breaks down all the hemoglobin into products. And then once the breakdown has happened, and once you have the product, the liver will transfer it to the gallbladder. Then when the gallbladder is going to release the bile, when the gallbladder is going to squeeze out the bile into the small intestine, all of the products of hemoglobin, or sort of the waste products of hemoglobin, they will also enter into the small intestine. Then from the small intestine, they will pass through the digestive tract and they're going to be excreted out in feces. So that is how hemoglobin is broken down. Basit, is that a bit more clear than before?
Yes, ma'am, understood.
All right. Now we are going to go over transport of oxygen. And this is something that seems simple, that we know how oxygen is transported. But now we are going to be looking at a lot of new things that actually we need to understand really well. So we have certain graphs that we need to know how to read very well. And we have got different types of hemoglobin that gives us different type of results. So we need to do that as well. So let's start with transport of oxygen. And you do get a lot of questions on this, by the way. So let's try to get our concepts clear so you can solve past paper questions easily. So when we talk about it, oxygen is of course transported by the hemoglobin that is actually present in your red blood cell. We have four protein chain. So we know that in the structure of hemoglobin, hemoglobin will have a quaternary protein structure, and it will generally have 4 protein chains. Each chain is so 4 protein chains that we have. And we know 2 of the chains are going to be your alpha chains, and 2 of the chains are known as beta chains. And on each chain, you are going to have your iron atom. One iron atom is going to bind to 1 molecule of oxygen. So since you have 4 iron atoms, one molecule of hemoglobin can carry 4 oxygen molecules. So hemoglobin has 4 chains and it has 4 iron atoms. That is why it is able to carry 4 oxygen molecules. Now, the reaction is a reversible reaction, and that is important. And I will tell you guys why it is important, but in a little bit. The reaction, of course, is that hemoglobin will combine with oxygen to give you oxyhemoglobin. Now, one interesting thing, when the first oxygen, so it can bind to 4, okay? So when the first oxygen molecule will bind to the hemoglobin, it changes the shape of the hemoglobin a little bit. It changes the shape of the hemoglobin a little bit. So it is easier for the second molecule of oxygen to come and bind. When the second molecule of oxygen is going to come and bind, again, the shape is going to change a bit. And it's even easier for the third oxygen molecule to come and bind. And the fourth one is going to be able to bind really quickly. And when you're doing the opposite, when you're breaking down oxyhemoglobin to release the oxygen, it will be exactly the opposite. The first molecule will come out really easily. The second molecule is going to come out a bit harder. The third molecule is going to maybe take a bit more time. And the fourth molecule is going to take the longest time to dissociate. So in binding, what is going to happen? Binding of the first molecule is going to take time. Then the second one is easy, third one is easier, and so on and so forth. And when you're doing the opposite, when you're breaking away the oxygen, when you are breaking away oxyhemoglobin to give away the oxygen, it's going to be the other way around. So when we are going, yeah.
Ma'am, in hemoglobin, each peptide chain can carry 4 molecules of oxygen?
Yes, each peptide chain, because each peptide chain has one iron atom. So one iron atom can bind to one molecule of oxygen. So one chain is going to bind to one molecule, the second chain will bind to the second one, the third will bind to the third one, and the fourth will bind to the fourth one. So each chain is going to be carrying one molecule of oxygen.
Okay, thank you.
All right. So when you are going to, now let's talk about your oxygen, how it is transported. We know that oxygen is we are going to breathe in, we are going to take in the oxygen. When we're going to take in the oxygen, we have our deoxygenated blood. So let me just go ahead and draw this. We have our alveoli inside our lungs. We have our alveoli, and with close to the alveoli, we have our capillaries. Now, what happens is that this is our red blood cells. What happens is that we know we have, so when we do the topic of respiration, again, we're going to do this in more detail, but generally what happens is that the oxygen, this is deoxygenated blood. This is deoxygenated blood. So over here, the concentration of oxygen is going to be low. Inside your lungs or alveoli, the concentration of oxygen is high. So what is going to happen, since you have such a vast difference in concentration, since you have the concentration gradient, concentration potential, so the oxygen is going to quickly be able to diffuse out from the lungs into the blood. After that, your blood is going to end up becoming oxygenated. Now, what happens in our capillaries is that the blood is that after the oxygenation has happened, so what is going to happen is that the oxygen is now going to enter into the plasma, and from the plasma, it is going to go ahead and enter into your red blood cell. When it is going to enter into the red blood cell, the oxygen will bind to the hemoglobin and it will give you your oxyhemoglobin. Now, if you look into it, where has the oxygen gone? To the red blood cell. In the plasma, if you talk about the plasma still has less oxygen. The oxygen has not increased in the plasma because whatever oxygen entered into the plasma, it diffused into the red blood cell. So your plasma still has lower levels of oxygen. That makes sure that the concentration gradient is maintained. That makes sure that the concentration difference between lungs and blood is maintained. And because that concentration gradient or because that concentration difference is maintained, what is going to happen? That the oxygen will keep on diffusing from the lungs into the blood, from the lungs into the blood. It will constantly be diffusing out, so oxygen can keep on binding to the red blood cells, and it is going to make sure that your maximum amount of oxygen is transported throughout the entire body. Making sense?
Yes.
Okay. Now we need to go over this particular concept. This is the graph for oxygen and hemoglobin. Saturation of hemoglobin, how much percentage of hemoglobin is saturated, how much hemoglobin or how much percentage of hemoglobin has oxygen in it. And on the x-axis, we have partial pressure of oxygen, how much oxygen is available, and depending upon the amount of oxygen, what is the percentage of hemoglobin that is saturated with blood. So generally, what you would say to me is that, Miss, the more oxygen you have, the more oxygen should be in hemoglobin. That makes perfect sense. The more oxygen in the body, the hemoglobin should be able to be more saturated. The hemoglobin should be attached to maximum amount of oxygen. So it should be a directly proportional relationship. It should be a linear line. It should be a directly proportional relationship. As the oxygen increases, the saturation of hemoglobin should also increase because more oxygen is available to bind to the hemoglobin. That is what you and I would assume. But when you actually look at the graph, or when you actually do your experiment and look at the results, you end up getting this sort of a shape. It is an S shape, and the curve that has an S shape, we call it a sigmoid curve. That's like the mathematical name of it, not just in biology. Mathematically, S-shaped curves are known as sigmoid curves. One thing we're going to observe is this. When you are dealing with low oxygen partial pressure, low partial pressure of oxygen, we see that the saturation with of the hemoglobin is also less. Yeah, that means when the partial pressure is going to be low, the saturation of hemoglobin is going to be less. Hemoglobin is going to attach to less oxygen. And when the saturation of it is going to be high, hemoglobin is actually going to attach to much higher level of your what do you call it, your oxygen, or when the partial pressure is going to be high, the percentage saturation of hemoglobin is going to be much higher. Now, what we can see over here, one thing that we need to observe and understand is this. Now, if it was a straight relationship, it was a directly proportional linear relationship, the percentage increase would be constant, right? From here to here, or from here to here, if you compared it, the increase would be constant. If it was increasing, for example, by 20%, this would also increase by 20%. The rate of increase would be constant, right? But that is not what actually happens when we are looking at our hemoglobin. The rate of increase of saturation is not going to be constant. So if you look over here, for example, a small increase in the partial pressure can cause a large increase in percentage saturation. What do I mean by that? Let's compare. This from here, this is your oxygen, or this is the partial pressure of oxygen. Let's take this as 2. And then from here. So look, it's a small increase, but that small increase has led to a large increase in percentage saturation. It is a small increase. A smaller increase has produced a large increase in percentage saturation. This is good for us. Why is this good for us? Because we want it, or we want that when you're going to, for example, okay, so the bottom part of the graph is what we observe in our tissue, in our cells. So when we're taking our blood to the cells, this is what we observe this is what we observe. And the high partial pressure is what we get when we are talking about our lungs. So the top part of the graphs for the lungs, lower part is going to be for our respiring tissue. Now, what is going to happen? Let's first of all, let's start with the respiring tissue. Okay. So when the blood reaches the rest, let's go over this first. Okay. Let's first talk about the respiring tissue. We'll talk about the lungs afterwards. In the respiring tissue, a small decrease in partial pressure causes a large decrease in percentage saturation. So a small decrease, you are just going from here to here. It is a very small decrease in partial pressure, but the decrease in the amount of hemoglobin saturation is very large. So a small decrease in partial pressure causes a large decrease in the saturation of hemoglobin. And that is what we want. We want that when the blood reaches the cells, when the blood reaches the tissues, that the hemoglobin quickly gives up all the oxygen. Where are we going to have less partial pressure? Inside the tissue, inside the cells. And we want that to happen. We want that even if there is a little bit decrease in partial pressure, even if the oxygen level falls down a little bit, we quickly want the hemoglobin to go ahead and give up the oxygen so that it can go to the tissue. And when you talk about the portion of the lungs, in lungs, what will you see? That even a small increase in partial pressure is going to end up causing a large change in the saturation. And that is what we want. That in the lungs, even if we are getting a bit more oxygen, we want the hemoglobin to, get more saturated so that the hemoglobin can, bind to the oxygen readily. It can bind to the oxygen easily. That is what we want. We want that in the respiring tissue, in the tissue that need oxygen, we want that even if there is a little bit decrease in oxygen, the hemoglobin quickly gives it up, right? And in the high pressure area or in the lungs, we want that even if there is a bit of increase in oxygen, the hemoglobin quickly binds to the oxygen in larger quantities. And that is why you're seeing this sigmoid shape. That is why you're observing this. Okay, look over here. Let me just show you in the graph as well. In the sigmoid shape, this is the respiring tissue, the lower portion, and this is the increase. Okay. Now, if I follow the sigmoid curve, look, the increase is much larger. If I do the same thing, but if I was following the linear relationship, if I was following the straight line, I would not end up observing it. I would not end up getting that. Okay, sorry. When you are if you end up looking over here, look, this to this, and from here to here. Now let's look at the changes. Okay, this is the change you are getting in the sigmoid curve, and this is the change that you are getting in the. Okay, hold on 1 minute. There you go. So what you can observe over here is that when we are following the linear line relationship, what are we observing? That even a small increase in your partial pressure, if you look at the sigmoid curve, you are getting a much larger difference. When you are going to look at the straight line graph, you will see that the difference is much less. This is why the straight line graph is what we would predict, but the hemoglobin actually has a sigmoid shape, because actually in the hemoglobin, a small increase in partial pressure will produce a large increase in percentage saturation. Even a small decrease is going to increase in your partial pressure will end up causing, just give me 1 second. So a small increase in partial pressure will cause a large increase in percentage saturation. And if you go ahead and you observe the same thing over here, oops, sorry. When you talk about your respiring lungs, you are going to go ahead and observe something similar. So you can see the difference between the pink lines, right? As if I follow the straight line graph, a small decrease is giving me a small decrease in hemoglobin percentage. But when I follow my sigmoid curve, I can see even a small decrease in partial pressure is giving me a large decrease in percentage saturation of hemoglobin. And that is what we want. We want that when we are at the lungs, that it is going to go ahead and it is going to, even a small increase in the partial pressure, even a small increase in oxygen will mean that the hemoglobin is going to grab as much oxygen possible and become saturated. And we want that when it goes to the respiring tissue where we need the oxygen, it is, even if there is a little bit decrease, even less oxygen, it will quickly give away the oxygen so that the tissue can get the oxygen. So this is what we get from this particular graph. Is this making sense?
Yes, ma'am. Got it.
Harsh, is this making sense?
Yeah, 100%.
All right. Okay, guys, so we are going to go ahead and stop it over here for today. And then from the next class, we're going to pick up from transport of carbon dioxide and continue from there.